So this is a second osmosis revision video. The first revision video was on the basics of osmosis, understanding how to describe osmosis and the terminology to use. This video focuses on the consequence of osmosis in cells. So as usual, there'll be a task to test yourself with. First, I just want to say that we will deal with animal cells first and then plant cells. So for these animal cells, the common example is red blood cells. So diagram of red blood cell, we'll refer back to that later. And what I'd like you to do is, for each of the solutions described on the left, I want you to provide a description of osmosis and then the consequence for the cell. And I want you to use the sentence structure we used in the first revision video. And it was something, well, it was this description basically. So in that we refer to solution A and solution B, or solution C, solution D, so on. Obviously you want to change that for this, and you want to be using this sentence structure. So refer to obviously outside the cell or inside the cell, because that's what we're discussing. So your task is to complete the table. Okay, so let's go through answers. Let's check how you did. So the external solution, if it's hypotonic, then hyper means high. That means there's a high solute concentration, and so it means that solution has a low water potential relative to the internal solution. So your description of osmosis should be this. The water potential inside the cell is higher than outside the cell, so net diffusion of water by osmosis out of the cell. The consequence for the cell is shriveling, or technical term crenation. There's a shriveled red blood cell. Isotonic, iso is same, so your description of osmosis should be this. So no net diffusion of water in or out of the cell. The consequence for the cell is no effect, so there's no structural change. And then finally, hypotonic. Hypo means low, low solute concentration. So you should have worked out that the description of osmosis should be this. So the water potential outside the cell is higher than inside the cell, so there's net diffusion of water by osmosis into the cell. What's the consequence for the cell? It's lysis, or bursting. And there's a burst animal cell. Okay, so I suggest that if you want to review this information, then maybe next time try and complete the table in a different order, just so you're not just remembering a pattern, so you're challenging yourself a bit. So maybe next time start with hypotonic, then hypertonic, then isotonic, just to test yourself. Okay, we're going to move on now to plant cells. So plant cells, so this is a little more information required. So first thing I want you to do is to label the plant cell. So there's some arrows there, or lines to particular structures. Please label those structures. So pause and test yourself on that. Okay, so you should have the cell wall. Remember, that's fully permeable, so everything can pass through. The cell membrane, it's a selectively permeable membrane. That's the vacuole. The tonoplast, which is the vacuole membrane, and then the protoplast. So the protoplast is the cell membrane, cytoplasm, nucleus, and tonoplast, and any organelles within that structure as well. So now I want you to complete the table. So you've got the external solutions on the left, description of osmosis, the consequences for the cell, and I also want you to describe what happens to the protoplast in each situation. So pause and test yourself. Okay, so we'll start with hypotonic, so low solute concentration. That means it's got a less negative water potential. So your description should be this. So there is net diffusion of water by osmosis into the cell. The consequence for the cell is turgid, and the protoplast is pushing against the cell wall. And remember, the plant cell doesn't burst because the cell wall is rigid, and so it's strong enough to push back. And this is a diagram of that. So there's no space between the protoplast and the cell wall. It's right up against the cell wall. Isotonic, iso, same solute concentration, so your description of osmosis should be this. No net diffusion of water into or out of the cell by osmosis. So the consequence for the cell is incipient plasmolysis. Incipient plasmolysis. What does that mean for the protoplast? It's beginning to pull away from the cell wall. So a diagram of that is this. So you start to see a small gap between the protoplast and the cell wall. Finally, hypotonic. So a high solute concentration, more negative water potential. So your description of osmosis should be this. Water potential inside the cells higher than outside the cell. So there's net diffusion of water by osmosis out of the cell. That results in plasmolysis. And so in that situation, the protoplast completely pulls away from the cell wall. So you've got something that looks like that. So you can see a very clear gap between the protoplast and the cell wall. Okay, so that's revision of osmosis in plant cells and animal cells.